Okay, we're back with this part 2. So we just got done talking about the first step of the biosynthesis of fatty acids. So we're going to continue on into the second step. So the second step, fatty acid synthesis from acyl-CoA and malonyl-CoA occurs by successive cycles of 6 enzymatic reactions. So just like the breakdown of it follows a specific pathway with specific enzymes, the synthesis also follows a very specific pathway. So in humans, specifically mammals, mammals, specifically humans, the process is going to be catalyzed by a fatty acid synthase multi-enzyme complex, whose sequence contains all the required enzyme activities, including an acyl carrier protein domain, so an ACP. We've seen ACP a couple of times. So just make sure that you remember what ACP stands for, and we'll keep seeing that pop up a couple of different places. Okay, so the intermediates in the fatty acid synthesis are attached to the acyl carrier protein with an addition of 2 carbon units at a time in one cycle. So similar to how we're breaking down the fatty acids by 2 carbon units at a time, we're going to be building it by 2 carbon units at a time. So we have our first reaction. It's the activation of fatty acid synthase by attachment of acetyl group to ACP or priming reactions. So this transfers an acetyl group from the acetyl-CoA to the ACP, and that is the acetyl-CoA ACP transferase. Again, remember, if we look at the enzymes, we kind of want to look at the more broad aspect of like what is actually happening, and that enzyme then is literally telling us exactly what is going on. Acetyl-CoA ACP transferase. So we're transferring an acetyl-CoA, we're transferring the acetyl group from the acetyl-CoA to the ACP, so it's being transferred. So the acetyl group is first attached to the ACP to form acetyl ACP. In the next step, the acetyl group is transferred to the enzyme portion of the FAS, which is FAS is the fatty acid synthase up here. Fatty acid synthase, also called the acetyl S enzyme. So that enzyme is the acetyl S enzyme, or the enzyme in the acetyl S enzyme is beta ketoacyl ACP synthase. That's like the longer name for it. Also looking at it, we'll see it called the KS. Again, it might be good as you're going through this to take all of the different enzymes and go ahead and write them in a specific place with maybe it's like some of their shortened abbreviations. Just as you continue to read and they start to use more abbreviations, it's a little bit easier to understand. Or you can kind of make yourself a little chart to go back and forth and see exactly what it is that we're talking about, if they do abbreviate. So the first reaction, reaction 2, is the activation of fatty acid synthase by the attachment of the malonyl group to the ACP. So remember, we have the acyl-CoA and also malonyl-CoA coming together. So this transfers a malonyl group from the malonyl-CoA to ACP. That second enzyme involved is the malonyl-CoA ACP transferase. And the malonyl group is attached to the sulfur group, the SH group, of 4-phosphopantothene, which is attached to the ACP. Thus, the SH group of 4-phosphopantothene is the site of entry of malonyl groups during fatty acid synthesis. That's the part that you'll want to remember, is the site of entry of malonyl groups during fatty acid synthesis. It's just saying that this is the side that the malonyl groups enter as we're building the fatty acids. Okay, so we have our ACP, pantothenic acid. This whole thing is our phosphopantothene. The malonyl groups are esterified to the SH group. So this is where the malonyl are going to be added. Okay, reaction 3 is a condensation reaction. We have decarboxylation of malonyl ACP, and then condensation of the decarboxylated product with the acetyl group using the third enzyme, beta-ketoacyl ACP synthase, or also known as acyl malonyl ACP condensing enzyme. So this, we have our acetyl S enzyme plus malonyl-CoA, and we get acetoacetyl ACP plus a carbon dioxide. Again, there's a lot of different enzymes and stuff here. I recommend, and a lot of AKAs. A lot of what you're going to see throughout the core science classes is that really there's just a lot of AKAs for the same thing. So it really helps to kind of keep a running tally. Excuse me. Keep kind of a running list of what all of the different AKAs are. So in this reaction, the acetyl group is transferred from the cis SH group of the KS. Remember, if we look at the different ones, KS was our beta-ketoacyl ACP synthase, also known as the acetyl S enzyme. So the acetyl group is transferred to the malonyl group on the 4-phosphopantothene SH group of the ACP. This acetyl group is becoming the methyl terminal 2-carbon unit of the newly formed acetoacetyl group attached to the ACPS group. Next, we have a reduction reaction. Following that, we have a dehydration reaction, and then another reduction reaction. So reduction of the beta-keto group by NADPH to form a hydroxyl group with this enzyme. The dehydration of the hydroxyl group to form a double bond. And here's our enzyme. And then the reduction reaction, reduction of the double bond by NADPH produces an alkane. So the double bond is an alkene. We produce an alkane using our enol ACP reductase. So production of the 4-carbon saturated fatty acyl ACP completes one pass through the FA synthase complex. The 4-carbon acyl chain is then transferred from ACP to the enzyme ketoacyl synthase. Now the ACP-SH site is free and ready to accept another malonyl-CoA. So we want to show you just real quick what the malonyl-CoA looks like. So the malonyl-CoA 1, 2, 3 is actually a 3-carbon structure, and this is what it looks like, 1, 2, 3. So we have the acetyl-CoA gets brought in carrying 2 carbons. The malonyl comes in carrying 3 carbons. But when we get the condensation reaction, and we have the decarboxylation of the malonyl ACP, we lose one of those carbons that was on the malonyl-CoA. So technically we're bringing in 2 from the acetyl-CoA and 3 from the malonyl-CoA, but one of the malonyl-CoAs gets condensated off and taken off, so we end up with 2 from the malonyl-CoA, 2 from the acetyl-CoA. Those combine and give us our 4-carbon chain. And then this process is going to repeat and cycle back through again and again until we get up to the size that we want. So that 4-carbon acyl chain is then transferred from ACP to the enzyme ketoacyl synthase. Now the ACP SH site is free and ready to accept another malonyl-CoA, and this completes 1 cycle. 7 cycles are required to synthesize a 16-acyl chain palmitate. And you think about it, that's because this, just like how when we were breaking them down, 7 cycles of breaking them down breaks down a full 16 carbon chain, because the very last cut gives you 2 and 2. When you're putting it together, the very first one brings 2 and 2 together, so you get a 4 chain from the very first reaction, from the very first time through, and then you're going to add 2 on over and over and over again from there. And this process consumes 8 molecules of acetyl-CoA and 14 molecules of NADPH. The final completed saturated acyl chain, palmitate, is then released by a thioester cleavage, which is an 7th enzyme, palmitoyl thioester. So what you're going to want to do, go back through this and pick out each of the different enzymes, and in your own words, put out what it is that each of those enzymes do. This is do within this specific reaction, because that's going to help you keep track of what each of the enzymes are, as well as better understand the actual process of what is happening here, to be able to answer questions about everything that's going on here. So we can see our fatty acid synthase. The carbon dioxide is coming off as we build the chain. So this right here is the overall reaction in 2 different parts. We have our first reaction, our second reaction, and the net reaction. And palmitate is the principal product of fatty acid synthesis and the precursor of other long-chain fatty acids. So we talk about the elongation of fatty acids. Once palmitic acid is formed, it may be elongated and unsaturated to synthesize other complex fatty acids. So we're going to build palmitic acid first before moving on to other stuff. The enzymes that are going to elongate palmitate are called elongases. So the elongase system is present in the smooth endoplasmic reticulum, you'll see it as SER, as well as the mitochondria. You want to remember these location questions because he will ask questions like that. The acetyl-CoA unit is added for chain elongation as the substrate for mitochondrial elongase system. So acetyl-CoA is connected to our mitochondrial elongase, whereas malonyl-CoA is the substrate for the smooth endoplasmic reticulum or microsomal elongase system. We want to keep those 2 separate as well. So acetyl-CoA for the mitochondria, malonyl-CoA in the smooth ER. It's the donor of 2-carbon unit is malonyl-CoA in the smooth endoplasmic reticulum or microsomal system. The donor of the reducing equivalent still is NADPH. Make sure you remember this, that it's a pH and not just an NADH, because the having the P there makes a big difference, specifically because of like where it's coming from and what its purpose and what its function is. Examples of fatty acids longer than palmitic acid. Palmitic acid produces stearic acid, which is an 18-carbon chain. The desaturation of fatty acids, which I might remember when we're talking about saturation, we're specifically talking about the hydrogens, right? How many hydrogens are bound to all the different carbons. So for example, palmitate can be elongated to stearate or stearic acid, and it can be desaturated and it becomes a different fatty acids, oleate. 18.1, the second number is showing whether or not it is desaturated, and you have the different double bonding. And this delta 9 is showing you specifically where that is that it's occurring, where that desaturation has occurred. So palmitate may be desaturated by desaturases to produce unsaturated fatty acids. Examples of two common unsaturated fatty acids are oleic acid and linoleic acid. I would highly, highly recommend that you keep track of some of these. Write them down as well, so that way you can start to keep track of the specific fatty acids. I'd write down the names of the fatty acids that we see here, as well as just the total number of carbons that are there, and then if it is fully saturated or unsaturated. We also have specific desaturase enzymes, so at 4, 5, 6, and 9 are located in the smooth endoplasmic reticulum. And desaturation of fatty acids requires molecular oxygen, NADH, and cytochrome B5. The enzyme is called fatty acyl-CoA desaturase, which is a mixed function oxidase, also known as a mixed function oxygenase. Double bonds present in naturally occurring unsaturated fatty acids of mammals have a cis configuration. So there's cis and trans configuration, so just cis configuration in mammals. In animals, double bonds can be introduced at the 4, 5, 6, 8, and 9 positions only, but never beyond position 9. This will be an important question for you. This will come up several times when you're being asked questions, okay? It is never beyond the position number 9. So we can have delta, let me make sure, 4, 5, 6, 8, and 9 here, delta 4, 5, 6, 8, and 9. But if you ever see delta 10, delta 11, delta 12, delta 13, anything like that, that is not produced in the body. And that will be an easy way to tell questions, to tell what are incorrect answers to questions or the correct answer depending on how the question is written. Examples of nutritionally non-essential unsaturated fatty acids. Non-essential meaning that our body can produce it. Palmitoleic acid, which is a C16 and has one double bond. The palmitic acid produces palmitoleic acid, 16-1 at the 9. Oleic acid from the stearic acid produces oleic acid, C18-1 at the 9 as well. Then our nutritionally essential unsaturated fatty acids. Again, these three you'll get asked a lot of questions about. The linoleic acid, the alpha-linolenic acid, and arachidonic acid. Make sure you write these down, put a star here. These you will get asked quite a bit of questions about during the next chunk of material. Okay, this last chunk, I'm going to go ahead and pause. I don't know, we'll just keep going. So we talked about the biosynthesis of the fatty acid chains, and now we're going to be talking about biosynthesis of triacylglycerols. If you remember earlier, triacylglycerols are the same thing as triglycerides. These are saying the same thing. Don't be confused. When you see both of these different words in different questions, they mean the exact same thing. And this is the simplest lipid formed from fatty acids. So the triacylglycerols are fatty acid esters of glycerol. Esters, we're talking about ester bonds. So these are essentially fatty acids that are bound to a glycerol. And it's a triacylglycerol because there are 3 fatty acids going to be bound to one glycerol, and that is going to make up our triglycerides. So fatty acids are incorporated into triacylglycerols for the storage of metabolic energy. So this is the storage. The triacylglycerols is one of the storage forms. Also phospholipids, components of membranes. Triglycerides are formed by the reaction of 2 molecules of fatty acyl-CoA with glycerol 3-phosphate. This produces phosphatidic acid. Phosphatidic acid is then dephosphorylated to produce diacylglycerol, or DAG. Then the diacylglycerol, or DAG, is acylated by a 3rd molecule of fatty acyl-CoA to finally produce a triglyceride. Reaction begins with successive acylation of glycerol 3-phosphate. So we have our phosphatidic acid, the enzyme here that acts on it, glycerophospholipid. This is our head group. So this is where we'll have bonding here. You can have it with just having 2, and this is a glycerophospholipid, or you can continue and have bound 1, 2, and then 3, and that is our triacylglycerol, or our triglyceride. So each of these R's right here is going to be a fatty acid chain coming off just like that. These are all the specific enzymes. A lot of this is this right here gets to be a little bit overwhelming looking at it just like this. At this time, I would take a quick pause, and we'll go ahead and actually pause this video here. Take a quick pause, and let's find another video. Again, I love good old Ninja Nerd. He's got a good one here. It's called Metabolism, the Triglyceride Synthesis. So I would jump over here, watch this. He'll take you through this specific process of making a triglyceride. And again, anytime that you're getting this is something you'll want to understand fairly well, because how triglycerides and how fatty acids and everything are going to start to form and be put together is going to come into play later on. So take the time to come through, watch this video. You can speed it up a little bit just to familiarize yourself a little bit more with this actual process. If you can read just these specific enzymes and these equations and like what it's going to, great, and just understand it from here, it's awesome. If not, go ahead and watch that video from Ninja Nerd. Really, really good. I'm going to go ahead and stop right here. Actually, I'll read this real quick. Lipogenesis, the overall processes of lipid biosynthesis, the conversion of fatty acids to triacylglycerols or triglycerides. Packaging of triglycerides into VLDLs. Those are very low density lipoproteins, and secretion into blood circulation. We'll talk more about this a little bit later on. This has to do with how triglycerides actually travel through the blood and get out to the different parts of the body that it needs to go to. Okay, I'm going to pause right here, and then we will continue just this last little chunk. There's just this little bit left. but I really want to make sure that I make its own little thing just to hit this by itself. Watch for that next video.